All right. Welcome back. Welcome back. Lab number 9, urinary system. What are we doing here? What are we doing? Let's see. We're going to look at the parts of the urinary system and the kidney. We're going to learn the parts as we trace the fluids through the nephron. We're going to compare the contents of blood compared to the glomerular filtrate compared to the urine. We are not going to do a urinalysis because we don't have that stuff. So we're going to skip that part. But we can still talk about what's supposed to be in there, what's not. And then we're going to look at the microscopic observation in the form of a slice of a kidney, and we're going to look at what the filtering units of the kidney are going to look like on the microscope. Specifically the glomerulus, that's what we're looking for, and the glomerular capsule. All right, so as we go through, we're starting off with some description, some functions, and starting with the kidneys. The kidneys are the organs of the urinary system, right? And the ureters, we have ureters on both sides. And what are the ureters doing? They transport urine from the kidney to the bladder, and it's at the bladder that we have temporary storage of urine, right? We have temporary storage. And when it's time for the urine to leave the body, it exits the body through the urethra. So the urethra transports, hit a button there. From the bladder to the outside. We do have a couple blood vessels. The renal artery brings unfiltered blood into the kidneys. Oh, my handwriting is not so great today. Where the renal vein brings, let's try to clean it up, brings filtered blood from the kidneys back to the heart. All right. Just remember the general rule here. The general rule is arteries away from the heart, veins to the heart. That's the general rule. So we can describe the functions of some of the parts here, right? So let's label these parts. Well, this area along the outer edge, it's slightly lighter in color. We call this the renal cortex. Now there's a thick kind of outer covering. This stuff out here, we don't have a label for it, but we could add it. This is called the renal capsule. It's pretty strong tissue. Going deeper, we see in here, we have these renal pyramids, right? These are the renal pyramids. But this whole region in here where we have the renal pyramids is called the renal medulla. And Just deep to that, we have the renal pelvis, and this is where all the collecting ducts would converge. It's in the hilus here that we have 3 tubes going in and out. They include the renal artery, which we described the function already. This brings unfiltered blood in, and then the renal vein brings the filtered blood out. And then we have the ureters. So if we were to describe each of these, the renal medulla with the renal pyramids, the renal cortex, this is the outer edge, the outer portion. This is the inner portion. If we look at these, this appears lighter. This appears darker. The pelvis is the site where the collecting ducts converge. And it's here where the urine would be leaving the collecting ducts and kind of funneling into the ureters. The ureter, did we do the function of the ureter on the previous slide? Yeah, we did. So we're not going to repeat that. We did these 3 on the previous slide. We're not going to write them again. The nephron is the basic unit of structure and function in the kidney. The nephron produces urine. And in the process of removing waste and excess substances from the blood. And we have about 1 million in one kidney, enough in one kidney to do the job. All right, so identify the parts of the nephron of the kidney model and give its function. Okay, so it's between the glomerular capsule and the glomerulus that we have filtration happening, right? So the glomerular capsule is going to receive the filtrate from that knotted group of blood vessels called the glomerulus. It's in the proximal convoluted tubules that the blood is going to be taking good things back. So this is the site of reabsorption, right? Remember, the cells in the proximal convoluted tubules have those microvilli that increases the surface area available for pulling those nutrients back. So that's all called reabsorption. The loop of Henle has a descending limb and an ascending limb. This loop of Henle actually extends from the cortex region into the medulla region. And this is a site of salt and water balance. Salts and waters could be exchanged between the blood and the urine here. And it's all the way at the very end of the nephron, the most distal portion of the nephron, that we see the blood dropping off larger items as a result of active transport. And of course, this is called secretion. The collecting duct receives urine from the DCT and sends it to the renal pelvis area. Pelvis. The renal artery, we did that one already. So these are vascular parts. So these are tubular parts, right? These are tubes of the nephrons, and these are the blood vessels. The renal artery, we said, brings the unfiltered blood into the kidney. It's the afferent arteriole that brings the unfiltered blood into those filtering units called the nephrons, specifically into the glomerulus. The glomerulus is a knotted or circular arrangement of blood vessels. Of capillaries. Remember, it's between the glomerulus and the glomerular capsule that filtration is happening, right? So this is actually let's add that. This is the site of filtration. Everything's coming out of the blood here. The efferent arteriole brings blood out of the glomerulus. So now the blood has been filtered. Glomerulus. And then that blood would end up in peritubular capillaries. So this carries blood over the proximal convoluted tubules, over the loop of Henle, and then over the DCT, distal convoluted tubules. And then that blood would be completely filtered. The items would be reabsorbed back into the blood that are needed. Secretion would happen at the very end. And then this filtered blood would leave the kidney through the renal vein, which we already did that one already. All right, so here we have a model, and we have a couple filtering units here, a couple nephrons here. Specifically, there's two that have labels pointing to it. So this is one. We can see the glomerulus is here, and the glomerular capsule surrounds it, the PCT. Loop of Henle, and then the DCT kind of brings the urine into the collecting duct, right? It'd be nice if we had just one. They kind of label some things on this one and label the other things on the other one. But the glomerular capsule is already labeled. And then from here, this would lead to the PCT, the proximal convoluted tubules. From the proximal convoluted tubules, the filtrate would then extend down the descending limb and then up the ascending limb of the loop of Henle. And then it would eventually end up in the filtrate would end up in the DCT, which they have labeled over here, before draining into this collecting duct. Now you can see one collecting duct receives urine from multiple nephrons. This is called the collecting duct. Sometimes it's called collecting tubule. What is this one calling it? Collecting duct. We'll stick with that. All right, so we have three main processes, right? That's happening. That they include filtration, reabsorption, and secretion. I like this picture here. It shows the unfiltered blood entering into the kidney, finding its way into the afferent arteriole, which is going to bring that blood into the glomerulus. As it spins around this circular or knotted arrangement, it's forcing the contents of the blood out and filling this space in this capsule that surrounds it, right? So this is going to be your filtrate. This is all filtrate. But a lot of that stuff ends up getting put right back into the blood. And then the blood leaves the glomerulus and it goes through the efferent arteriole before it goes into the peritubular capillaries. And this would be this would follow the loop of Henle and then eventually the DCT. But what's happening here? We got three major processes that happen. The first one is filtration. Where does this happen? Where does filtration happen? It happens between the glomerulus and the glomerular capsule. So what is the direction of the movement of materials? Well, we have two choices for that. From the blood to the nephron or from the nephron into the blood. So in this case with filtration, everything's taken out of the blood. So it's from the blood to the nephron. I said everything's getting taken out of the blood, but we know it's not everything because some of the things are just too big. Red blood cells are just too big. They're not going to leave the blood. Large proteins would be too big. They're not leaving the blood. But what is water? Salt, nitrogen compounds like urea, glucose, amino acids, vitamins, minerals, maybe. All right, this brings us to our second process, which is reabsorption. Of course, this happens in the proximal convoluted tubules. Now we see items moving from the nephron back into the blood. And what would we want to send back in the blood? Well, glucose, we don't want to lose glucose in our urine. We don't want to lose amino acids. We need those. We've got to build proteins with those. Perhaps maybe some vitamins, we could put those back. Maybe not. Secretion would be the last step, and this is happening at the distal convoluted tubules. And this is when we take things that are still in the blood that we couldn't get rid of earlier because they were just too big. So from the blood into the nephron. And these are larger contents. For example, drugs. Either recreational or prescription. All right, so let's pretend that we are a molecule of urea. Urea is a nitrogen waste product. And we need to be removed from the body. And right now we're floating around in the blood. So there's urea, that's us, we're in the blood. The blood enters the kidney and it enters the kidney through they're giving us a hint here. Through the renal artery. And then we're going to end up from the renal artery finding our way into an afferent arteriole and then finally into the glomerulus. It looks like the vascular components of the nephron are written in red. Vascular meaning the blood vessels are written in red. From the glomerulus, we're leaving the blood vessel and entering into the tubular portion, specifically starting with the glomerular capsule. So now we're not in a blood vessel anymore. Now we've left the blood. We're going to be urine. Not quite urine yet, but we're getting there. From the glomerular capsule, we go to the PCT, the proximal convoluted tubules. And that's going to send us through the loop of Henle. And then into the DCT. And then the urine leaves the DCT and drains into the collecting duct. From the collecting duct, it goes down through the renal pelvis into the ureter. It gets stored in the urinary bladder. Until finally it's time to leave the body. And when we exit the body from the urinary bladder, we have to go through the urethra. All right, so which blood vessels carry rich in oxygen? Well, those are arteries. Which blood vessels carry less in that's the opposite, isn't it? But which blood vessels carry the blood that has a lot of waste? Well, the renal artery brings the unfiltered blood in, right? And then less waste. The renal vein leaves the kidneys because the kidneys have just filtered the blood, right? Makes sense. All right, so this is what you should be doing on your own. I'm not going to do this for you. I'll do the first one for you. With water, there's 180 liters of water that are filtered by the kidneys every day. That's a lot. Does that mean we pee 180 liters every day? No, because most of that water, as it's removed from your blood, just gets put right back. All right. So how much? Well, 180 liters were removed. 2 of that actually leaves as urine, but so most of that gets put back. So we're going to get mostly absorbed. I'm going to put an M here because M means mostly absorbed. Glucose, 162 grams of glucose was removed from the blood by the kidneys, but guess what? 0 grams end up in the urine. So that is totally reabsorbed. Totally reabsorbed. Keep going with this. See what happens with salts, amino acids, proteins. If you were to do a urinalysis, you would pee into a cup and then would dip a urinalysis test strip. And they would change colors depending on what's in there and what's not. So let's say that there was blood in the urine and there was proteins in the urine and there was glucose in your urine. Well, this would be a very bad urinalysis test strip to get, because with blood in the urine, that means you have red blood cells, and this would be an indication of hypertension. Unless there's broken down red blood cells, if you have hemoglobin in your blood, in your urine, that indicates bleeding in the upper respiratory tract. Proteins would be another indication of hypertension. And the reason why these two are both telling us that there's high blood pressure is because they're too big to really leave the blood. You know, the pores in those glomerular capillary walls are small, so really the plasma goes through and only the tiniest things are really leaving the blood. However, if these larger items are leaving, they're getting forced through those walls due to the high blood pressure. Glucose is an indication of diabetes. The specific gravity is a measure of how much solute is in a solution. So if a solution is more concentrated, it's going to have a higher specific gravity reading. Water is as dilute as it can be, right? So water has a specific gravity reading of 1.000. However, if you add solutes to the water, that specific gravity reading can go up. In urine, that is very concentrated would have a specific gravity reading, something like that. However, when would the urine be concentrated and when would it be dilute? Under what conditions will the specific gravity of urine be at the low end? So this is when you are hydrated. If something is at the low end, this means that it is dilute, like water. As opposed to being dehydrated, then the specific gravity reading would be at the at the higher end, right? So when you are dehydrated, when you first wake up in the morning, you aren't drinking all those hours that you were sleeping. Your body's dehydrated. Your urine is going to come out concentrated. Oh, forgot the H. You know I can't leave that like that, right? All right, and of course, pH is a measure of how acidic things are. And the normal range of for urine, the pH would be somewhere between 4, which is acidic, to 8, which is alkaline. All right, so that's the normal pH range. Again, we have the cortex is the outer edge. Inside that, we have the medulla. And then we have the renal pelvis. And that's a ureter. All right, let's look at this microscopic observation. We're going to look at the slice of the kidney under high power. And I want you to estimate the diameter. Oh, I can't leave that like that. One glomerular capsule. And of course, I want this answer, as always, in micrometers. Some of you have been giving me on the lab exams measurements in millimeters. No bueno. So let me pause the video. I got to pull up the PowerPoint that shares this microscopic image with us. So I'll be right back. All right, so let's pull this up. I have it here. And we're going to scroll down to again, this is always a good review. We got our models here, cortex, medulla, and then we have the renal pelvis, the artery, the vein, and the ureter. This is the real one, cortex, medulla. This medulla has the renal pyramids in there. The renal pelvis is where everything converges down into. All the nephrons are up here in the cortex, but the collecting tubules converge down through the pyramids, and then everything drains into the pelvis, and the pelvis delivers that urine out the hilus down and through the ureters. This is right out of the book. We see in the cortex, that's where we're going to find all the glomerular capsules and the glomeruli, so the beginning stages of where filtration is happening. And then we have those loops of Henle that extend down into the medulla region. And the collecting duct, this is another model that would be on the tables if we were in school, so we could see the renal arteries, the afferent arteriole, the glomerulus, the efferent arteriole, the peritubular capillaries kind of going all over the place. Renal vein. Renal vein brings the filter blood out. Now, this is what we're looking for, but this is under low power. So these are the glomeruli. Here's one, here's one, here's one, here's one. And you see this clear space that surrounds. Let's get a bigger view. That's the glomerular capsule. And this is what I want you to measure is the diameter of one of these. So you're going to be drawing one of these to scale using this image. That's the glomerulus. This is the knotted arrangement of cells. All right. And then we see the border of the glomerular capsule is marked with simple squamous epithelial cells. So flat, plate-like epithelial cells. All right. And every dark purple circle is a nucleus. So there's a lot of cells here. All right, so this is your microscopic observation. Make sure you are labeling each of those things here, because parts to be labeled: the glomerular capsule, the glomerulus, and the simple squamous epithelium that line the outer edge of that glomerular capsule. Put your lenses in and your magnifications in, and then estimate the diameter. All right, and that's it. So, yeah, any questions? You know where to find me. Well, if you have any questions, you could go to the discussion board and put them there. But if you have any other concerns or questions, you could also email me. All right, bye-bye.